Yeah, so I am in immunology, so I study MS, as Dr. Gaubat said. But in this talk, we're going to talk also about a variety of diseases other than MS, but mainly about MS and EAE, since that is really where we know I have a lot of information. It's always, all right. So when we talk about autoimmune diseases, these are diseases that are caused by an adaptive immune response to some self-component, typically self-proteins, but antibodies can also bind to lipids and carbohydrates and DNA and all sorts of things. But so when we talk about adaptive immune response, we're talking about either B cells and the subsequent antibodies that they produce, okay? Being reactive to self and mediating some sort of pathology in the context of autoimmunity, or mediated by T cells. And T cells, there's a couple different major types. We have T helper cells that primarily their role is actually to help other cells of the immune system do their job. Or there can be CD8 T cells, and CD8 T cells mediate their pathology predominantly through lysing targets. So they have their receptor recognizes some sort of self component and kills the target cells. That's very brief, and we're going to get into a little more detail. So these are the diseases we're going to talk about today. And again, most of the talk will be about MS and EAE, which is an animal model used to study MS. But I don't want you to have the impression that there are no autoimmune diseases in the CNS other than MS and EAE. And I have question marks on all these human diseases because the reality is we know a lot less about them than we probably should. And some of them, there's even some question as to whether they're true autoimmune diseases, but there's increasing evidence that they are. All right, so this is the first one. I actually have some videos, which I'm going to try to make this work. Actually, I'm going to switch my screen sharing to a different screen and see if I can get this to work better. Let's see. Is this going to work? No, that's not the right screen.
Amy, you're being pretty daring this morning, huh? I wouldn't try to do that. Nothing ever works for me.
I'm all set up.
I'm Sam Barr, and I have stiff person syndrome.
Oh, here it is.
I want to do a video. I'm feeling pretty good today, but I want to do a video of me walking because I'm going to be getting braces tomorrow.
Can y'all see this one? The video playing?
This is the before video.
Let's go back to my original screen, my other one. Okay. All right. So that gentleman had stiff man syndrome, which is, well, I'm telling you the technology is still driving me crazy. So this is a relatively rare disease. The incidence is about 1 in a million. Typically occurs in young adults, which is another key that is probably autoimmune, because most autoimmune diseases occur in young adults. My cursor now doesn't want to work. So these patients have these severe muscle spasms that actually cause their legs to be rigid, and other limbs too. You see it in the lower limbs pretty obvious, and you can see in this picture it's actually an 11-year-old boy who has it. So it can occur even in younger ages. So in stiff man syndrome, it appears that these patients have antibodies to GAD, which is the enzyme that converts glutamate to GABA. And GABA is an inhibitory neurotransmitter. So by inhibiting the production of GABA, you end up actually getting continuous signaling that's causing this rigidity that you see in these patients. The other thing that's kind of interesting is GAD is not expressed in many tissues. It's expressed obviously in GABAergic neurons, and it's expressed actually in the pancreas. So there is some association between this rare incidence of stiff command syndrome and patients who have type 1 diabetes. In type 1 diabetes, patients also have antibodies to GAD. And this is basically showing antibody staining of the pancreatic islets, showing abundant expression of GAD. And you also see this in the cerebellum. Now, it's interesting because even though there is a high incidence of type 1 diabetes in patients that have Stefman syndrome, it doesn't go both ways. And there's certainly no 100% between the two of them. And it turns out that when they've looked at the antibodies that are generated and the specificity of the antibodies, meaning how they bind, the epitopes that they bind, are often different, which is why you don't see this 100% overlap between patients who have type 1 diabetes and patients who have stiff man syndrome. So this is another one. So this, let's see if I can, oh, that seemed to work. Okay, so here is a little girl who has Sydenham's chorea.
There, you got it.
Dr. Amy, I think the video screen is not being shared. Not sure.
Can y'all see it
Put
or
the
not?
marks, put the cap down, sweetie. You don't need to hold on to the cap.
Not really working. Y'all can't see that screen. Okay. All right, let's try to, I'm really. Am I still on? Can y'all hear me?
Yes.
Okay, because I can't, yeah, so I guess y'all can't see that, the videos that I'm showing.
We saw the first one.
Oh, you saw the first one, but you couldn't see the second one. Okay, I'm going to skip the rest of the videos. This is getting too complicated. So the video was a little girl, and if you saw her, she just kind of has this continuous movement, like, her arms are just kind of flowing around. Sometimes they call it kind of a dancing syndrome, because the children, and actually it can occur in older people, it's more common in children, but this can occur in older people as well, but they have this kind of continuous arm movement, leg movement, body movement, as if they're kind of dancing. And these involuntary movements are actually caused by a condition secondary to having beta hemolytic streptococcal infection. This is the infection that actually causes strep throat, okay? So another complication of strep throat, a more common complication of having strep throat or beta hemolytic streptococcus infection, is called rheumatic fever. And what happens is you get this strep infection, most commonly strep throat, as most of us know. You generate an adaptive immune response, particularly a strong antibody response. And some of the antibodies that were directed against the bacteria actually cross-react with components in the heart. Also, they can affect actually joints as well. And so they develop what's known as rheumatic fever. And this occurs probably about 1 in 100,000 cases, where Sydenham's chorea is more about 1 in a million cases. And in Sydenham's chorea, what you see is that these antibodies, again, generated against strep, actually bind to either lysoganglioside or the dopamine receptors, causing upregulation of dopamine in dopaminergic neurons, and again, causing this kind of continuous firing of the neurons, so that you see this continuous kind of uncontrolled movement in these children. The good thing about anytime you have a disease that's mediated by antibodies is that you can do plasmapheresis, where essentially you can filter a patient's blood and remove the antibodies from the blood, and they're kind of removing the source of the problem. And so most of the time, people with antibody-mediated autoimmune diseases, can typically get under control fairly easily. But if you had seen the child, it's a little girl, cute little girl. If you saw that in your child, it would be pretty traumatic for the parent anyways. But the child actually looked quite happy. But again, most of these things are, kind of traumatic. They're fairly easy to cure. So another condition is neuromyelitis optica. And this is actually a disease in which patients generate an antibody to aquaporin 4. And aquaporin 4 is a water channel that's expressed on astrocytes. So these patients, they generate these antibodies. The antibodies bind to the aquaporin 4 on astrocytes, causing dysregulation of a loss of the aquaporin 4 function. That's a water channel. This results in also a loss of function of the excitatory amino acid transporter 2. And this transporter is important for the uptake of glutamate by astrocytes. And when you have so you end up with this excess glutamate that increases excitotoxicity, the overactivation of postsynaptic neurons, and actually upregulates inflammation and damages myelin. Okay, so you've got this whole cascade of events that all originate by this antibody to aquaporin 4. Now, what causes the induction of an antibody to aquaporin 4 is a little unclear. But again, this is another case where it's thought that it's possible that actually an immune response to an infection may have triggered the production of an antibody that cross-reacts with aquaporin 4. Another thing that's kind of notable about NMO, sometimes you'll hear it called neuromyelitis optica spectrum disorder. So the name comes from the observation that these patients, the inflammation is typically in the spinal cord, affecting motor function, or in the optic nerve affecting vision. In this case. And most patients, previously to the recognition that there was this antibody for aquaporin 4 in these patients were all originally diagnosed with MS, because if you look at the lesions, if you look at the pathology in the spinal cord and the optic nerve, you see a rather complex inflammatory response. One of the notable differences between the inflammation that's seen in NMO versus MS is the neutrophil. And neutrophils are abundant in NMO. And these are, of course, innate immune cells that are prevalent in infections as well. You do not see neutrophils in MS lesions. So if you have a patient that presents either with like optic neuritis or with lower limb weakness, and a lot of times they're trying to figure out what's going on. They do a spinal tap, and if the immune cells come back with neutrophils, that kind of gives them a hint that it's NMO versus MS. But it was really the identification of this aquaporin 4 antibody in these patients that now we have a cause for NMO that is distinguishable from what we see in MS patients. All right, so transverse myelitis. So this is inflammation in the spinal cord. And in these patients, there's a single lesion. When I say lesion, the size of the lesion can vary significantly from being very, rather long versus fairly compact. But it basically encompasses the entire width of the cord, which is again a bit unusual that you see an inflammatory lesion that is that extensive. So these patients again have a tremendous amount of lower limb weakness and paralysis. In about 50% of the patients that present with transverse myelitis, they can say that they've had some sort of infection within the past month or so. This can sometimes associated with viral infections. It can be bacterial, although viruses are more common. But one of the reasons this has become a lot of conversation in the news is the fact that in the AstraZeneca trial for COVID-19 vaccine, they had a patient actually develop transverse myelitis. Transverse myelitis, again, it's quite rare. About 1 in a million people will develop transverse myelitis in their life. So it's relatively rare. But so there's a lot of concern with, yes.
Is this position,
Yes.
is that permanent?
Hello?
Yeah, can you hear me?
No, it is not permanent. So most of, can you hear me?
Yes.
Can you hear me? Yes, am I back?
Yes.
Okay, good. My thing just said unstable internet. I'm in my office, but OSU internet. Okay, so yeah, so typically cover because of the demyelination and inflammation that occurs, but most patients do recover over weeks or months in time. But again, in this is another case where this is, again, about 50% of the time seen post-infection. The other 50% in other autoimmune diseases, and commonly seen in patients with MS, where they get this extensive inflammatory lesion. Now, the inflammation can look different depending on infection versus a transverse myelitis associated with MS. So again, in MS, you will not see these neutrophils, but if it's post-virus, you also see the neutrophils. And the loss and the damage to myelin can be attributed to antibody production and mediated by complement damage. It can also be mediated by inflammatory mediators and like cytokines, reactive oxygen species that are produced by like microglia and macrophages and things at the site of the inflammation. So the patients are treated with robust anti-inflammatory drugs to reduce the inflammation and allow for remyelination to occur. So this is a model that was originally developed. It has a kind of a long history, actually back to the rabies vaccine. So this is Louis Pasteur, who developed the rabies vaccine. The rabies vaccine was rather crude. Essentially, he would inoculate rabbit brains with virus, then take the brain and spinal cord from those infected, homogenize it, set it out in the sun for a couple of weeks to let it bitten by a rabid dog, and the boy recovered. So from that vaccine, there would be a occasion where someone who had received the vaccine would develop an insist due to the fact that the virus had not truly been inactivated, because rabies virus obviously infects the CNS, and was it just a complication of rabies virus that was not inactivated? Or was it something about the fact that you're giving humans this spinal cord homogenate? So, a physician in the early 1930s actually started looking at this, and actually then took immunized animals with either virus or just the CNS tissue alone. And what he found was that there was the same incidence of this encephalomyelitis, whether the virus was present or not present in the CNS tissue. So from that, he also noted that this disease looked a lot like acute disseminated encephalomyelitis, which is an inflammatory demyelinating disease seen in children. It's a monophasic disease. So they get this onset of these neurological symptoms, pretty robust, But the kids recover again in weeks to months, fully recover. So, so EAEs have evolved a lot over the many decades people have been working on it. And now essentially we mostly use mice. Almost every rodent, monkey, guinea pigs, all sorts of things have been used for EAE. Now, of course, we mostly use mice. And if you immunize mice with a myelin protein, even myelin peptides now, depending on the strain of mice, in complete Freund's adjuvant, these mice will develop a paralysis. And it's typically an ascending paralysis, meaning the tail's involved first, and you'll see hind limbs affected. You can see bladder incontinence and even some forelimb involvement in these mice. And if you look at the inflammatory lesion, it looks very similar to the lesions that are seen in MS patients. The other thing we know is that if you take the draining lymph nodes from these mice where the T cells have been activated, you can culture those T cells with the myelin protein in culture and take just the CD4 T cells and transferring them back into a mouse. And that mouse will get the same exact disease, EAE disease. Okay, so this allows us, and today we actually, in my lab, have, and lots of labs, we have even better tools because essentially we can engineer CD4 T cells that have a T cell receptor that recognizes myelin. So now you can essentially, you don't even have to immunize, you take the T cells from the spleen to figure out how encephalogenic T cells are developed and transfer those into mice. Can you still hear me?
Yes.
Okay, because my internet keeps warning me there's a problem. Okay, good. Okay. So these videos aren't working. So this is a mouse with EAE. So when you pick up a mouse by the tail, the first thing that should happen is the tail should stick straight up. And you can see this mouse has curled tail. So that's basically showing that the limp tail. And if you look at this mouse on the left, you'll see the mouse is on the ground. So mice usually run around with their tail up, not dragging the ground. And the other thing is you look at his foot sticking out here. And that's because typically mice have their feet under them when they're running around. And so this is called a wide base. So they spread their legs out to become steady to compensate for their weakness. And humans do the same thing. You'll see patients often will spread their legs out and things to make them more stable when they're weak. So there's basically the symptoms of EAE and MS are caused initially by the inflammation. So this is looking at in the brain. This is a mouse with EAE. This is a blood vessel. And you can see these inflammatory cells crossing the blood-brain barrier into the brain. This is a control mouse. And if you take myelin-specific T cells and you label them with a green marker, a green label, you inject them into the mouse, you will see that those myelin-specific T cells will traffic to the central nervous system. This is also, again, in the brain, and the red is myelin. And you will see they will make their way to myelinated axons, and you can even see where the myelin is being damaged by these cells. So how did we figure out it was CD4 positive T cells? Well, I kind of told you already how it was done originally, but essentially when you immunize the mice, initially they transferred serum, which contained antibodies, and you transfer serum, you see no induction of EAE. You can sort into different populations by transferring B cells, CD8 T cells, CD4 T cells, and of course EAE is only induced when you see the transfer of these CD4 positive T cells. So again, showing that they, in the case of MS, are in EAE, not MS, are T cells. And initially, this was predominantly done by Th1 cells, so that you could transfer Th1 cells and you see this beautiful response. Today, there's a lot of work being done on Th17 cells and their role in EAE and in MS. If you read a lot of literature now, you would think that it's a Th17 mediated disease. I can assure you from research done in my lab, as well as many others, that it is Th1 cells are just as effective at inducing EAE. And there's a lot more evidence in MS that Th1 cells are pathogenic actually than Th17 cells. So this is still up for a debate, but it definitely appears that that Th17 cells, even though that's very popular in the literature right now, that is not the sole story. So in EAE, to get that robust response, the other thing that's important to remember is that you have to induce innate immune cells, or this danger signal, to actually get the T cells to actually become activated. So that's the role of the complete foreign adjuvant. And CFA contains mycobacterium. And when you inject, if we just give myelin antigens, you will not induce EAE, okay? In fact, you'll probably actually induce tolerance. But when you put it in with the mycobacterium, essentially what you're doing is you're allowing for dendritic cells and macrophages in the skin to actually will become activated because they recognize components of mycobacterium as being foreign. And it's that initial activation of those dendritic cells and macrophages that are really important, because then they phagocytose the myelin antigens, process them, take them to the draining lymph nodes where the T cells that will become activated to the myelin antigens that were in the adjuvant or with the adjuvant. So if you don't have the adjuvant, you don't get that initial robust danger signal and that phagocytosis, and that allows for those cells to move to the lymph node where they will interact with T cells. Okay. So again, as I mentioned, it's, we know that it's the CD4 positive T cells. So in EAE, we're inducing the disease. So we know what the cause of the disease is because we're immunizing them, we're activating the T helper cells that then cross. And once these activated T cells actually cross, they cause this blood-brain barrier breakdown, lots of inflammatory cells will cross. So actually, when you look in the lesions, what you'll see there's plenty of B cells. There's lots of CD8 T cells. You'll see macrophages there. You'll see microglia that get activated, and a robust production of inflammatory cytokines and other inflammatory mediators. Now, that being said, we know that antibodies, even though they are present, and actually, there's a lot of antibody production actually in the CNS and MS patients, that it doesn't appear that the antibodies themselves are a major contributor to the pathology. And that most of it is mediated by, the pathology is mostly mediated by inflammatory mediators actually produced in response to the CD4 T cells interacting with microglia and macrophages in the CNS. All right, so the next step is demyelination. So obviously that inflammation is causing, and those inflammatory mediators are damaging myelin. And you can see, this is actually from a mouse with EAE, a lesion in the spinal cord. The blue dots are the inflammatory cells that have come in. Red is a myelin stain, which you can see is absent here in this lesion. And again, that compromises, the loss of myelin is compromising axonal conduction. And where the lesion is obviously dictates what the ultimate symptoms of will be. So demyelination, because myelination actually promotes saltatory conduction. So you have the myelinated regions of an axon, you have the nodes of Ranvier where there are voltage-gated sodium channels, and the action potentials essentially jump from node to node. And this is very, very efficient. So in MS or in EAE, you get this myelin damage. And obviously that's going to compromise axonal conduction. It also, even though it compromises axonal conduction, there are, the sodium channels have the capacity to redistribute. So usually within a few days of a patient having an onset of a new inflammatory lesion and the onset of new symptoms, often within a few days, they'll say, I'm starting to feel a bit better. And that's usually because of this redistribution of sodium channels. So when you redistribute them, now you can restore some conduction along those axons. It's not the same as if it was myelinated, but it is some restoration. The good thing is that we have the capacity to remyelinate. So typically what happens over the next weeks or months is you actually get remyelination in this area. The sodium channels get back to the nodes of Ranvier and the patients will recover. So that's called a relapse and a remission. So they relapse, they have new onset of symptoms because of this inflammation and the loss of myelin. But as you get restoration of the myelin, the patients will recover. The problem really comes in is when you have this permanent damage and to the axons. So the axonal damage. So axonal damage is permanent. So once the axons become severed, they will not repair. And any neurological deficits that have occurred now, instead of being able to recover because of remyelination, now these become permanent. So this is looking actually from MS patients' pathology, and the green is a neurofilament marker. And so these are neurons, and you see these bulbs. And these bulbs are areas where the axons have actually been severed. So you're basically, the transport of things along the axon gets halted, and they start to accumulate, forming these bulbs. If you look at this one, the red is MBP, so you can see that this axon has been damaged by myelin, and you can see, and then it was severed. So this usually happens when patients, often the inflammatory, you get this relapsing remitting course going on, but often the same areas will be vulnerable to inflammation. So you'll get say optic neuritis, you'll have inflammation in the optic nerve, you recover over the next few weeks, and a lot of times the patients are given steroids to help minimize the inflammation, but a year later you get another optic neuritis in the same place. So when you have this inflammation that occurs repeatedly in the same place is when you increase the probability that you're going to get these axonal severings and this permanent deficit occurring. So I'm going to skip video. So MS, so this is the most common neurodegenerative disease of young adults. It typically is diagnosed in individuals between 20 and 40 years of age. The incidence in the United States is about 1 in 1,000. And women are about 3 times more likely to develop it than men. Again, this is kind of very common of autoimmune diseases. They are typically more common in women and usually diagnosed in young adults. The cause of MS is unknown. So if you just look at the pathology again, you'll see these scarring, these sclerotic plaques where you get the names. This is showing around the periventricle area. It tends to be an area where demyelination occurs quite frequently. You can see it there. And again, before the aquaporin 4, patients with NMO were diagnosed with MS. There's another population of individuals that were previously thought to have MS as well. And this was a group of people that had a HTLV1 infection, which is a retrovirus in the HIV family of viruses. It was called HTLV1 associated myelopathy or tropical spastic paraparesis. And so these patients, again, developed this inflammatory demyelinated disease that looked very much like MS, pathologically indistinguishable. And before we discovered HTLV1, these patients were all diagnosed with MS. So one of the things is MS is kind of a diagnosis where you're ruling out lots of other things and going, okay, we've ruled out this and that, and we're left with this. Whether it's a single disease or just a family of disease that have a common phenotype, people are still trying to figure out. Okay, but again, the components, major components in EAE and MS are basically the same. If you look at the course of MS, the typical most common course is what we call a relapsing remitting course, where you have this onset of new symptoms and recovery. And this happens intermittently. Some people, it happens every year or two. Some people can have exacerbations like once a decade. Some people have them, maybe several in a year. So this varies a lot. But almost all patients will switch from this kind of classic relapsing remitting to a progressive phase in which they have which we call the secondary progressive phase, where in addition to these relapsing, they're continuing to progress neurologically, accumulate neurological deficits, even without the obvious induction of new inflammatory lesions. In the paper that we're going to talk about, the clinical trial is kind of dealing with patients in this realm. So there's a lot of medications, anti-inflammatory medications or immune modulator medications that are designed to help patients in the relapsing remitting phase, but very little about what we can do to stop this progressive phase of the disease. The other thing that's not on here is that I'm going to show you, this is brain volume. So the other thing we know is that as patients, and with aging, our brain volume goes down very slowly. In MS, this brain volume decline occurs at a more rapid rate. And so this is another way that we can measure, or that is being used in clinical trials to measure therapeutic efficacy. Can you change the rate of loss of brain volume? So that's going to be in that paper as well. The other thing is that there is another type of MS that is much more rare. Only about 5% of patients have this, and that is patients who don't have these inflammatory, these relapses. They kind of had this very gradual accumulation of neurological deficits, and they have evidence of inflammation in the CNS, but they don't have these acute relapses. And again, it's kind of curious, these patients that are diagnosed with what we call primary progressive MS, meaning they just kind of have this gradual decline or accumulation of neurological disability. You know, is that the same disease? Or are we just calling it MS because it has neurodegeneration with inflammation? And that's still up for debate. But those patients are also included in the trial on the paper. So it includes all progressive. So trying to figure out what the cause of MS is, there's been a lot of epidemiological studies done. And some of the very interesting data, it basically shows that people, the further you live from the equator, so up here in the northern hemisphere, or way down here in the southern hemisphere, your risk of developing MS is higher. And if you migrate as a child from a higher latitude, say you live in Canada, and then you move to Texas, you actually, as a child, you actually acquire the same risk as a Texan. But if you move as an adult, your risk is the same as from the region you came from. So this kind of suggests that there's something to do with being at higher latitudes that may affect risk. And so sunlight, and there's a lot of evidence that is a risk factor in adults. So the same people settled in Canada, in the Northern United. The other thing is infections, which comes up in autoimmune diseases as well. And that the infections that are common in, say, colder northern climates are a bit different than the types of infections that people have who live near the equator. And some immune response to an infection may be contributing to that risk. So there's a story about the Faroe Islands. So this is a group of islands, actually, in here, you don't even see it. It was actually a very isolated set of islands that were settled by the Vikings. And so the genetic background of the individuals are the same as the people from Norway, okay? And we know, and Norway had a high incidence of, and has had a high incidence of MS as far as records show, okay? But there was no evidence that anybody on the Faroe Islands had ever had MS, okay? And again, this was a very isolated set of islands prior to World War II. But during World War II, British soldiers came and occupied the Faroe Islands. And after the war, they left. In 1950, the first case of MS was documented in the Faroe Islands. So about 5 years after the soldiers had left. It was a lot of work. They thought this was going to be the perfect way to figure out what the cause of MS was, because again, you had no MS, a very isolated population, genetically high susceptibility. And so they thought it had to be an infection. So there was a tremendous amount of work done looking for a pathogen that the British soldiers brought. But actually, they can never definitively say or identify a pathogen that seemed to be associated with the outbreak of MS in the Faroe Islands. And to this day, the Faroe Islands has the same incidence of MS as Norway and Northern Europe. So the other thing that's kind of interesting is that MS patients have myelin-specific T cells. That shouldn't surprise anybody, but so do all of us. And if I take peripheral blood and analyze the frequency of T cells, of myelin-specific T cells in almost anybody's blood, whether they have MS or don't have MS, the frequency is the same. And this has always kind of been mind-boggling, because if you believe that myelin-specific T cells are the cause, wouldn't you expect to see a lot more of them in patients with MS versus healthy individuals? But we don't. So there's been so this was really kind of a conundrum. So a couple experiments done actually in the 1990s actually kind of address whether the T cells in MS patients were fundamentally different than those in healthy individuals. Mainly, were there's any evidence that the myelin-specific T cells in MS patients had ever seen myelin and reacted to myelin? So were they memory cells or effector T cell phenotypes? And this seemed like it should be really easy, but it actually, really was a little bit of a challenge. But a couple actually simple experiments kind of solved this. So one group did this by looking at mutation, taking myelin-specific T cells and looking at the number of mutations in the HPRT gene. Now, any of you who do like real-time PCR or lots of other gene expression studies know that you use this as your control, because the level of HPRT is kind of continuously expressed at a baseline level all the time. So in cells that have proliferated a lot, meaning they've gone from, say, being a naive T cell to an effector T cell to a memory cell, where they've actually seen their antigen and proliferated a lot, you would expect an increase in point mutations, right? Because every time cells proliferate, they make minor mistakes. So this study basically looked at the mutation rate in the HPRT gene in myelin-specific T cells that came from healthy individuals, or myelin-specific T cells that came from MS patients. And basically, they found an increased mutation rate in the myelin-specific T cells from MS patients compared to controls, suggesting that those T cells had proliferated more often than the myelin-specific T cells from MS patients. So this was one of the first pieces of evidence that said there was some difference between those cells in a control and an MS patient. So my lab did a study that came out just after that one, and this was looking at how T cells become activated. So naive T cells need two signals to actually become activated. The T cell receptor must engage the myelin peptide in the context of MHC. We call this signal 1. And you have to have this co-stimulatory signal between CD28 and B7. And both of these signals are needed for a naive T cell to proliferate and become activated initially. So we looked at whether, but at the time, it really wasn't fully known, but we suspected in our lab from some other studies we had done that this signal was not needed for effector or memory T cells. So we looked at what would happen if we blocked this and stimulated T cells to myelin, whether we would see a difference between healthy individuals and MS patients. So this is looking at, so this is increasing proliferation, and this is our control, so no antigen. This is stimulating with myelin basic protein or tetanus toxin. So everyone should recognize tetanus toxins because everybody gets their tetanus shot every 10 years, so you should have a robust response, okay? And this was our control. And this was when we blocked that CD28 molecule, and this is in an MS patient. And what you see is that you see this response to MVP, and even when we block that co-stimulation molecule, we see that response, okay? Suggesting that that is now a memory or an effector response. And tetanus, robust both places. And this is just another MS patient that shows the same thing. But if you look at what we saw in healthy individuals, again, this healthy individual has this fairly robust response to myelin basic protein, a good response to tetanus. But when we block that CD28 molecule, the myelin basic protein response goes back to baseline. It's gone. So these myelin specific T cells are naive. They've never seen their antigen. So this was one of the first studies that actually, again, showed that fundamentally there's something different about the myelin specific T cells between MS patients and healthy individuals. Now, that being said, we don't know, that doesn't make them necessarily pathogenic, because it could be that in MS patients, the T cells are getting activated in response to the illness, but not necessarily causal clear. A variety of clinical studies were starting to be done to actually figure out, how interesting, because a lot of them necessarily didn't teach us how to treat them, but we learned a lot about the disease from doing these clinical trials. So one of them was done, and this was done actually in the late 80s, I believe, was to target the CD4 T cells. What happens if we get rid of them? And you think about when we were EAE, this was done. They treated that would deplete CD4 T cells, and the vast majority of mice, this ameliorated their symptoms. This is the treated. These are the number of animals that did not have symptoms. And you can see with severe symptoms, the EAE, but once you deplete the CD4 T cells, the mice with severe EAE is dramatically reduced. So this study was actually done in humans as well. Again, this is in the 1980s. So they treated patients with a CD4 antibody that caused CD4 T cell depletion. And this is looking at the number of CD4 T cells in the patients after treatment. These are the placebo-treated patients. These are the ones with the CD4 depleting antibody. And you can see there's a nice depletion, but the CD4 counts aren't gone. They're low, but not completely gone. If you look at, this is looking at the number of active MRI lesions. These are the CD4 treated patients, so they have fewer active MRI lesions compared to the placebo. Now, it turned out that this was not statistically significant. And you can see, even though these have fewer, they still have, they're still moving along and still having inflammatory lesions. So the great thing about this study was that there was a lot of work to look at the CD4 T cells in these patients pre and post treatment. And what they found was the number of interferon gamma positive T cells. So these are saying the Th1 cells, these effector cells that make interferon gamma, they didn't do such a great job of getting rid of them. These were the patients treated, this is the placebo. So even though the total number of T cells went down fairly significantly, they didn't get rid of all of them. And the cells that remained were the cells we thought were probably pathogenic. So they reduced the number of pathogenic cells, but didn't get rid of them, which probably was reflected in the change, the improvement, but not really a major difference. Now, after this study, this strategy was dramatically dropped because, as I said, this was going on in the 1980s. The other thing that happened in the 1980s was the onset of HIV, which of course causes CD4 T cell depletion due to the virus and has significant health consequences. So this kind of, so from this we learned we really shouldn't be going, this is not the right strategy because of other serious consequences. All right. So then the next strategy was, should we target the cytokines that these T cells are making? Okay, so at the time, Th1 cells were clearly pathogenic in EAE. MS patients clearly appeared to have these Th1 cells as well. So the thought is, well, if we just target interferon gamma, the primary cytokine produced by Th1 cells, will that be beneficial? So in EAE, there was a surprising response. They actually made mice deficient in interferon gamma. They made mice deficient in interferon gamma receptor. And those mice all had EAE still. And in some cases, even more severe EAE than actually the control mice, which is this line here. And so that made people really a little bit leery and like, what's going on if it's not the cytokine being produced by the Th1 cells? But obviously, T cells have lots of functions beyond a specific cytokine being produced. This was actually a human clinical trial that was done giving patients interferon gamma antibodies. And this is looking at the number of patients that have not had any progression, okay? So this is at month 1. This is the black one is the patients that got the interferon gamma antibody. The white is the placebo. And the gray is it's an antibody for TNF alpha, another inflammatory cytokine that was thought to be important in potentially mediating pathology in MS. And what you can see is the patients who received the interferon gamma antibody actually did best in this clinical trial. But because of the data in the AE, there was a lot of hesitancy to move forward with interferon gamma blocking strategies in MS. So continuing to move on, and this is just showing that data. So for interferon gamma, the patients without any new enhancing lesions was almost 87% compared to the placebo was 40. So, you know, even though this was kind of impressive, this is basically where it ended. So the next thing was trafficking. So what if instead of like worrying about the CD4 T cells or their cytokines, we just stop them from getting to the brain? So there's a variety of strategies that have been used. I'm going to talk about one of them. There have been others to modify trafficking, and there is quite a bit of success with trafficking. So it was known that for lymphocytes to actually breach the blood-brain barrier, they have to be activated for once initially to initiate that initial blood-brain barrier breakdown. And that they actually use VCAM on the endothelial cells interacting with an alpha-4 integrin actually on the T cells to actually initiate this migration. So a drug was developed, which was an antibody that was specific for alpha-4 integrin that would prevent the alpha-4 integrin on the lymphocytes from interacting with VCAM. So this was done in mice first, and in mice it works beautifully, and so it was then done in humans. So this is from a clinical trial for natalizumab, this anti-VLA4 therapy or alpha-4 integrin therapy. This is the number of new lesions that patients are generating over time in 6 months. This is really short. And this is the placebo. And yet you can see in the 2 doses of natalizumab, it almost completely shut down any formation of new lesions. I mean, so the blood-brain barrier basically closed. People thought this was fantastic. This was by far at the time the most successful clinical trial. And the fact that you could get this data in 6 months was also unheard of, because MS is kind of a slowly progressing disease. So the fact that you could see these changes so robustly so early was really impressive. The problem was a couple months after the drug was FDA approved, there was a patient who developed multifocal leukoencephalopathy. Now, this is a fatal, almost always fatal disease caused by reactivation of JC virus. Most of us never heard of JC virus before this drug came out. The JC virus is a common virus that is latent in neurons, and about 65% of Americans have JC virus. So basically this drug was put back on the market, and now if you're JC virus positive, you will not be given this drug for fear of this PML occurring. So another piece of evidence in MS patients that that MS is a true autoimmune disease targeting myelin was a study, again, originally originated in EAE, called altered peptide therapy. And what happens in this is in EAE, we have a very good idea of like exactly what peptides, you know, bind to the MHC to stimulate T cell receptor to generate these encephalogenic myelin-specific T cells. So knowing that, you could actually make modifications to the peptide, which would allow it to bind to the MHC, but in a way where the T cell receptor no longer recognized it. So you kind of bind up the MHC molecules, but the T cell couldn't bind. So in EAE, where all this is very well defined, you're using inbred mice that all have the same MHC, this worked beautifully. So they decided to do this in MS. So they made an altered peptide ligand to actually treat patients with. There were a couple mistakes in this trial. One was they didn't select patients based on their MHC. And you have to remember that peptides bind to MHC. You know, there's a lot of specificity in the ability of a peptide to bind to MHC. And they didn't consider that in the trial, which was a big mistake. So this is some data from that altered peptide ligand. This was in the phase 1 trial. So this was the first one. So this is one patient 4 weeks after they into their APL therapy, and this is their MRI. Okay, normal, normal, normal. Pretty much, maybe there's a little something right there, but mostly fairly normal. This is a week later when the patient started to have new onset of symptoms, and these are all these new inflammatory lesions you can see here. Okay. So this was really unfortunate. So this patient went from having no lesions or maybe very little evidence of lesions to having dozens. It turns out this patient had dozens of new active lesions. And so this APL trial again, actually this patient had 91 new gadolinium enhancing MRI lesions, 91. I mean, so this trial, needless to say, this happened in several patients and was stopped immediately. And so this, instead of this myelin basic protein, this altered myelin peptide inhibiting disease, it actually caused activation and robust disease in several of the patients in this trial. So again, more evidence that myelin actually is contributing, or T cell responses to myelin are contributing. I'm going to see what I'm going to do this real briefly. So one of the problems is, and this is just a few therapies, we have a lot more, and almost all of them deal with that early inflammatory phase of the disease, where you're basically trying to stop inflammation. And if we stop inflammation, we can stop demyelination and stop neurodegeneration, right? The problem is there's a lot of patients that already are past that initial relapse remitting phase, or that or even not responding well even to anti-inflammatory drugs, that are still having disease progression. So there's new work trying to figure out how do we stop the neurodegenerative component, or kind of that innate inflammatory component. So it's thought that progression, that activation of microglia is still continuing in these patients, even without significant T cell infiltration coming in. So, so we had done a study in mice to target NOGO-A. So NOGO-A is an inhibitory protein that's expressed in the adult CNS, and it prevents axonal growth. So during normal development, you don't have NOGO-A, and that allows your axons to develop normal and myelinate. And actually, as they myelinate, that's when NOGO is expressed, because it's expressed by oligodendrocytes. And it's the upregulation of NOGO-A and some other inhibitory proteins that actually stop axonal growth. And this is important in normal development, that you get a normal growth of axons that make appropriate connections. But in the context of MS, the problem is that if you need to, if you have axonal damage, one of the reasons they can't sprout and repair is that you have remyelination occurring, and you have upregulation of proteins like NOGO-A that inhibit the axon. So you've got these two opposing forces going on in the lesions that are detrimental. So we actually generated an siRNA to NOGO-A to see if we could down-regulate NOGO-A in the lesions and whether that would promote axonal repair. So this is just looking at NOGO is in red. This is in a cell culture, and you can see when we give the siRNA for NOGO-A, we had about an 80% reduction in the amount of NOGO-A expressed in the cell line. We then tagged the siRNA with a fluorescent tag and injected it IV to look at where it went. And if you look at these two cells, these two cells are larger, and you can see that the fluorescently tagged NOGO siRNA gets into the cytoplasm where it needs to be for an siRNA. And this is looking at EAE and two different models of EAE. And essentially, we treated mice shortly after they got sick, and about 2 weeks later. This is the siRNA NOGO, and you can see that these animals recover quicker and do better long-term than the control animals. In this model, this is a different model, same idea. We treated once they started getting sick, and you can see that the ones that got no-go A actually did better long-term. And I don't know if, and we did have evidence that we saw decreased no-go in the mice. So this is looking in the controls at the border edge. And this is looking at no-go here compared to here. And you can see there's a significant downregulation of no-go. We also saw upregulation of GAP43, which is expressed on growing axons, which we saw that as well. All right, and then to the paper. Okay, so this again is a phase 2 clinical trial. And again, I like doing clinical trials because it's something as basic scientists we don't teach a lot about. And I think it's really important that basic scientists know what's going on in their field and kind of understand how drugs get to that point, how the work that we do is translated into drug development, and how clinical trials are conducted, and how we analyze the outcomes. Okay. So the paper is about a phosphodiesterase inhibitor, and it's a phase 2, so that's an early trial. And the reason this is interesting is because phosphodiesterase play roles in how T cells function. It plays roles in how oligodendrocytes function, neuronal plasticity, astrogliosis, so a whole bunch of targets are potentially that are relevant in MS can potentially be modulated by inhibiting phosphodiesterases. So we're going to talk about that. And again, the paper in many regards is really simple, but I want you to think beyond. I want you to think about how they did the trial, whether what they measured were good measurements for outcome, what other things they could have looked for clinical relevance. All right, any questions? And we can talk about questions on Thursday as well, because I will be there. Or if you have questions now, I'm happy to.
Great, thank you, Amy. Anybody have questions?
Dr. Lopet-Raki, I really enjoyed your presentation. I just had a couple of questions about like some of the earlier diseases that you talked about before MS. For stiff-neck syndrome, it seems that the limbs were kind of in that kind of extensor position. So I just kind of wanted to know if like the loss of GAT, if it like specifically affected the extensor muscles over the flexors, and like or is the GAT loss just everywhere?
Right. And so I'm not a neuroscientist, so but this has to do with, so there's different types of neurons. So you have GABAergic neurons, which are primarily affected in, say, stiff man syndrome, where you're taking glutamate, converting it to GABA, which is an inhibitory. So when you lose the inhibitory neurotransmitter, then you get this kind of continuous action through glutamate, which is an excitatory neurotransmitter. So it's really in a subset of neurons that this is occurring, and I believe primarily in the cortex. It affects about 30 to 40% of neurons. But the other thing that you kind of remember when you look at these neurotransmitters, so again, there's a variety of neurotransmitter. Different neurons use different neurotransmitters to different degrees, and you have the excitatory and the inhibitory, right? So there's all these balances. And so one of the reasons I like showing the videos of these different neurological diseases is like some of them you would look at the patient, they look very similar, and yet some of them look very different. So if you look at the man with stiff man syndrome, you see this very rigid, because you're basically having this uncontrolled transmission that's causing this rigidity, right? Unfortunately, if you had seen the girl with Sydenham's chorea, where you have where the dopaminergic neurons are the ones affected, it looks different. You actually see, you have this, again, you're having this excitatory pathway that's getting continuous, but instead of having rigidity that you see in that patient, you actually see this continuing flowing movement. So this is all about different types of neurons and whether the neurotransmitter that's affected is either inhibitory or excitatory.
All right, thank you. And I think my last question was for transverse myelitis. You said that the inflammatory lesion could be more compact or more widespread. What's kind of mediating the width of the lesion?
Right. So that's a good question because so by MRI, sometimes I could have shown y'all different MRI images. You'll see these very kind of compact. And what makes it transverse myelitis is really the fact that the entire width of the cord is affected. The why you have, I think that whatever the underlying condition that's causing the transverse myelitis probably is directly related to the size of the lesion. Okay, so you know, and I don't think anybody really knows, and it also could be the point at which, you know, because depending on where the lesion initiates. So if initially it's like something causing, say, tingling in your right foot, but that tingling in your right foot on day 1, by day 3, if it's my right leg doesn't move at all, that goes from being a very small lesion, you know, in your lower cord to seeing now potentially a very large lesion that's moving up the cord by the time you get the more severe symptoms and you go to the doctor, where that has a pretty dramatic effect. You're more likely to seek medical care, and therefore it gets taken care of before you see a much larger lesion. So there's a lot of factors that probably play into the lesion side, including whatever the underlying condition is, as well as when patients seek medical care for it.